All right, it's my pleasure to introduce our next generation leader, Jennifer Garrison. Jennifer received her Ph.D. with Jack Taunton at UCSF, and then did a postdoc also with Corey Bargman at Rockefeller. She's now an assistant professor at the Buck Institute for Research on Aging, and you must be very busy because you also hold appointments in the Department of Cellular and Molecular Pharmacology at UCSF and the Davis School of Gerontology at USC. She is an Alfred P. Sloan Neuroscience Research Fellow and received a Glenn Foundation Award for research in biological mechanisms of aging. Jennifer's interest in neural circuits and neuromodulation, I think, began with Corey, where I wanted to mention one story I found particularly interesting of where you found an evolutionary precursor to the famous neuropeptides oxytocin and vasopressin called Nematocin, and proceeded to demonstrate a role for this in a circuit that involves mating. Your interest in the relationship between the anatomy of circuits and neuromodulation really continues in your lab to this day, where you're now studying both nematode and mouse, I understand, and also how neuropeptide neuromodulation encodes long-term changes that influence both behavior and aging as well. So welcome. The title of her talk is Cellular and Circuit Mechanisms of Neuromodulatory Signaling. Thank you.
Thank you for that kind introduction. I'm actually going to talk about nematocin a little bit in my talk today. So I first want to thank the organizers. I can't imagine how much work went into putting together this whole showcase. I had an amazing set of meetings yesterday, and I'm very much looking forward to today and tomorrow. So the major focus of my independent lab is to try to understand how the behavioral outputs of a neural circuit are assembled from the diversity of transmitters that they express, and in particular to understand how neuropeptides influence behavior. So obviously as neuroscientists, we want to try to relate the structure of the brain to its function. But I think that a major challenge in doing that is that the functional structure of the nervous system is not fixed. So it can be changed by the action of chemical neuromodulators. And I think that this is really exemplified in C. elegans. So as you mentioned, Ed, I work in both worms and mice. But the first story I'm going to tell you is about worms. So on the right is a picture of the C. elegans connectome. So this is the only animal for which we have the entire brain map, all 302 neurons. I think this is the most beautiful representation of the data, although it's not very data rich. Each one of these points is a cell, and the lines between them are the connections, either synapses or gap junctions. And we've had this map for almost 30 years, and we still can't understand how the behavioral outputs of this very simple animal are encoded by the nervous system. And what we'd like to be able to do, obviously, is to interpret it like an electrical circuit, to have the map, excuse me, and then to be able to predict how information will flow through it. So this is another way to represent that data. And on the left here is a picture of all 302 neurons in the worm lit up with GFP. And I think that one of the reasons that we can't predict how behavior is encoded just having the anatomical map is that superimposed on the anatomy is a separate map of biochemicals, these neuromodulators that can change the functional output of the system without changing any of the anatomy. So I'm coming back to the more beautiful picture of the connectome here. But this is a picture that we're all very familiar with, and what this connectome is depicting, for the most part, are the chemical synapses, and we're very familiar with how fast neurotransmission works at chemical synapses. Small molecule neurotransmitters, as Evan mentioned, the 4 humors, and a few others, but not very many more than that, are released in response to an action potential. The signaling is directional, it's very fast, it's confined to the synapse. And these neurotransmitters are very quickly either degraded or recycled back into the presynaptic terminal. And so this kind of communication is it's very much linked to the anatomical map. But the class of neuromodulators that I want to talk about today are neuropeptides. And so this is the largest and most diverse class of neuromodulators. And neuropeptides, unlike small molecule neurotransmitters, are not confined to the synapse. So they can be released to the synapse in response to an action potential, but more often you find them outside of the synapse and released in response to other signals or different signals than what we find just action potentials. And what this means is that because neuropeptides are not restricted spatially by the wiring diagram or temporally by rapid degradation, they can signal over relatively long timescales and potentially long distances, although this is really an open question, to coordinate neurons that are not anatomically connected. And so this is the map that I'm interested in figuring out. And with neuropeptides, the situation gets a lot more complicated when you consider that there are hundreds of peptides encoded in both mammals and in worms. And one neuron will typically encode multiple peptides, and they might have overlapping or antagonistic functions. And one peptide can bind to multiple receptors. And really, since they're signaling through G-protein coupled processes, they could theoretically signal to any neuron that expresses the appropriate receptor. And because, of course, G-protein coupled processes are very slow relative to ionotropic channels, trying to correlate what's happening with the release of the peptide and then figure out what cells it's signaling to is very difficult in the context of, say, a mammalian brain. And that's one of the reasons that we're using worms. There are some questions that we haven't been able to address in mammals that we'd like to find the answer to. And so humans and worms have about the same number of neuropeptide genes and receptors, and they share a conserved set of enzymatic pathways that are responsible for the processing and the transport and the exocytosis of peptides. So this is a signaling system that evolved really early, and we think that this is a reasonable place to study some of the cell biological questions that we're interested in. And I probably don't have to convince this audience that neuropeptides are important, but I will say that there are a lot of peptides in the cortex, a lot. And we really don't know what they're doing. And part of the reason I think that we don't have a better picture of what's happening is because we don't understand at a basic level, at a physical level, how these molecules are signaling. And so I'm not going to go through this list, but what we're thinking about is how there are really probably three maps, at least, of brain function. One is the anatomical map that we all know about. The other is, of course, activity, which can be superimposed on that. And the third is the flexible modification of the nervous system by these neuromodulators, and that's really what we're interested in. So I've organized my talk into two parts, really. I'm going to start with an example of one neuropeptide and how it can control behavior in worms. Then I'm going to move on to tell you about the work that we're doing in the lab in both worms to look at how neuropeptide signaling is specified in space and time, and then to move on to what we're doing in mice. So hopefully I'll get through all of it. So you probably don't know in the audience that when I was a postdoc in Corey's lab, I worked on a project that it was basically based on a discovery that Evan McCosco made. So it's nice to have him in the audience for this talk. And what Evan did was he found that the C. elegans genome encodes an ortholog of the mammalian peptides oxytocin and vasopressin. And neuropeptide genes are notoriously difficult to annotate, and that's because they're very small. They tend to have very few coding exons. And so Evan was able to find this peptide by taking advantage of this unusual domain that's contained in the proprotein precursor called a neurophysin domain. So most peptides are cut out of larger precursors, and they don't contain this neurophysin domain. And what's beautiful about that is it's very well conserved, so he was able to pull it out bioinformatically. And he also identified two potential receptors that have homology to human oxytocin and vasopressin receptors. So Evan moved on to bigger and better things, as you just heard. And because I was really interested in this idea that peptides can modulate behavior, I decided to follow up on this discovery. And so I showed that the peptide does actually bind to these receptors in a heterologous cell system. And I spent a little bit of time actually figuring out what the active peptide was. There was a little bit of ambiguity at the amino acid level. Oops. Go back there. There was some ambiguity at the amino acid level. There were multiple potential processing sites in the precursor. And so coming from a chemistry background, I was very excited to grind up worms and do a lot of biochemistry. And in the end, we confirmed the actual structure of nematocin using tandem mass spectrometry. And what we found is that it's a little bit longer than oxytocin and vasopressin, but it maintains a lot of the same chemical features. So that's nice. We've figured it out at a molecular level, but we really wanted to know what it was doing in behavior. And we saw this as an opportunity to really look in detail at what a neuropeptide was doing in a circuit. I'm not going to go through this. You all know why oxytocin and vasopressin are important. So with basically just using really straightforward genetic analysis, using mutants in either the peptide or the receptor, I put them through a battery of assays. They were very much normal in almost every assay we looked at, except for mating behavior. And this was a surprise to us. We didn't really expect to see this behavior to be conserved so far back in evolution. But what we found is that if you knock out either the peptide or the receptors, that when you put a male worm with a hermaphrodite in a context where he should be mating, about half the time they fail. They just don't end up mating. And you're going to learn more about worm sex right now than you really ever wanted to know. But what's, one of the real strengths of C. elegans, aside from the genetic tools, are the ability to really take apart a behavior and quantitate every part of it and then relate it back to the genes. So mating behavior in worms can be divided into both long-term and short-term behaviors. If you put a male worm on a plate by himself and there's no hermaphrodites around, he'll actually execute what turns out to be a very long-term behavior for a worm and go searching for a mate. And what we found is that with worms that were lacking the nematocin peptide or the receptors, they were less motivated to find mates. So that was interesting. And looking at a much shorter time scale, copulation in worms is much like reproduction in other animals. There's a sort of courtship where the male worm will execute a very stereotyped series of motor programs in order to transfer sperm. So I'm going to tell you about this in a little detail. So the male contacts the hermaphrodite through interactions with its tail. And once he contacts the hermaphrodite, he starts backing up. If he comes to the end before he finds the vulva, he'll execute a very sharp ventral turn. Then once he finds the vulva, he'll transfer sperm. And you can quantify a lot of aspects of this motor program simply by putting a single worm on a plate with receptive hermaphrodites and making a movie and analyzing it. So this is a worm porn. Yeah, that's right. So this, the worm that's moving here is the male. This is a wild-type male. There's the tail and there's the head. And this is a hermaphrodite. And what you'll see is that very quickly, he contacts the hermaphrodite, makes a turn, finds the vulva, and will be transferring sperm in a moment. So this typically with a wild-type male, they'll stop to mate with the very first hermaphrodite they find. They'll transfer sperm within a few minutes. And it's a very efficient process. So let's put this. Now, the movie on the right is a Nematocin mutant male. And I've started this movie about 10 minutes in. And this male he's come into contact with 8 other hermaphrodites before he decided to stop and mate with this one. And what you can see is that he's having a lot of trouble. He's clearly motivated to try, but this movie will go on for quite some time, and he will never actually successfully transfer sperm. But if you're watching, what you can see is that he contacted the hermaphrodite. He's backing up just fine, and he's making turns, he's scanning for the vulva. So all of the individual components of this motor program are intact in these mutants. And I've collapsed a lot of data onto this one slide because I wanted to get to the punchline and move on to other things. But these are ethograms where each line is a single movie of a male mating assay. And what you can see in the top 10 lines, these are the wild-type worms. What we did is we broke down the behaviors into sub-steps, and we've looked at very fine detail at what's happening and color-coded it. So if you just ignore the details and look at the overall patterns, what you can see is that in the wild-type males, they very quickly and efficiently go through the motor program and end up transferring sperm. And of course, on the individual level, there's variability, which is what we always see in behavior. But there's a progression that ends up, in all cases, except for this one, with sperm transfer. And if we had extended the x-axis out here, we would see that this worm also was able to successfully transfer sperm. Whereas in the mutants, you can see that all the colors are there. So all the individual motor programs are there. but they're completely disorganized, and the transitions between them, so the transitions, generally you see the color go from blue to green to yellow here. The transitions between them are disrupted. And so what we found essentially is that the peptide is providing a global organizing role. And this is what we think is a fundamental property of peptides, that it's not required for mating behavior, but that it's telling the neurons that are involved in this behavior to progress in the right order, basically giving them a context cue. So we can look at this, we can basically break this down into productive or non-productive behaviors, and you can see that in wild-type worms, they're biased towards productive behaviors, whereas in the mutants, essentially, 50% of the time they're doing productive things, and the rest of the time they're not. And so we took this information and we related it back to the expression patterns that we determined for the peptide and the receptors. So one of the beautiful things about only having 300 neurons is that you can really ascribe function to individual neurons. And this has been done for mating behavior by many, many labs. And I've highlighted a couple of the neurons here that we know are important for different substeps of these behaviors. And I've color-coded the expression pattern of the peptide and the receptors. This is a close-up of the male tail. And so what we find, actually, is that the expression of the receptors and the peptide makes a lot of sense with what we know about the function of these neurons in mating behavior. But we wanted to know what the relevant site of expression was for this particular behavior. So we used CRISPR to remove the peptide from subsets of neurons that were in the endogenous expression pattern. And so DVA is a neuron in the tail that expresses the peptide. And when we removed the peptide from DVA, what we found is that release from DVA seems to mediate most of the behavioral responses that we saw. And if you take a step back and you look at where DVA is in the wiring diagram, we think that DVA is releasing nematocin to encode this sort of slow arm of the mating program, while these other neurons are mediating the fast classical neurotransmitter responses. And that nematocin is essentially providing an instructive signal to these neurons to help them interpret sensory information that in some contexts could be taken for aversive things, and really telling the neurons that these sensory cues in this context are meant to be good and should progress to the mating program. And importantly, DVA is not synaptically connected to any of the downstream neurons that express the receptors. So this is certainly something that's happening outside of the normal wiring diagram. So the model that comes out of this is not surprising. We were pretty happy to find that the expression pattern of the receptors fit with what we knew about mating in the male tail. But what was surprising, as I mentioned earlier, is that this behavior is conserved through evolution. And this is something that we see often with neuropeptides, that they're locked into some sort of behavioral function early. And as evolution proceeds, the details change. So human mating behavior and worm mating behavior are very different in terms of the substeps. But the peptide providing this global organizing role may be actually very similar. So I'm going to switch gears now and talk about what we're doing in my lab. And I don't have time to talk about everything. And I'm very happy to talk outside of this seminar to anyone who wants to hear about what we're doing. But in the worm, we're really looking at the cellular level. We want to answer these basic questions about how neuropeptide signaling is functioning. What we want to do is we want to understand, with the same clarity that we understand fast transmission, we want to understand how neuromodulation is happening by neuropeptides. And so these are the sorts of questions we're asking. We want to know, what is the function of this release outside of synapses? So this is a very difficult thing to address in a mammalian brain and especially in an attacked brain, because axons and dendrites are often interspersed. We want to know what's the wiring of the biochemical circuit. So we can take the expression patterns for peptides and receptors that we know about in the worm and bioinformatically look for relationships between how they're spread out, right? We know they're not synaptically connected, but is there some pattern that can emerge from how many synapses are between them or how far they are in distance. We want to look at how signaling changes over time. And particularly, we want to look at how signaling is terminated. So we really don't understand how these signals are turned off. And we think that this might be one mechanism for specificity in these kinds of neuromodulatory circuits. So peptides are, they're transported in large, dense core vesicles, which are very different from the clear vesicles at axons that are that contain classical neurotransmitters. And we're taking advantage of just this nematocin, oxytocin, vasopressin specific domain called the neurophysin domain. So neurophysin is required for the peptide to be loaded into the vesicles. It's thought that the neurophysin domain protects the peptides from intermolecular disulfide bond exchange. You've got 10,000 of these molecules in a vesicle, and they all have a disulfide bond, so you could easily get a big mess. But what's lovely about this is that they're not covalently linked once they're inside the vesicles. So we can tag the neurophysin and in that way label specifically these vesicles that have the nematocin in them. And this is important for us because we're operating under the assumption that there are multiple distinct, functionally different populations of vesicles, some that could access, say, the axon and the synapse, and others that might only be able to access the soma or the dendrites. And what we'd like to do is to find specific markers for these subpopulations of vesicles so that we could then genetically manipulate them and figure out what is the function of different types of vesicle release. And of course, we can also use a pH-sensitive GFP. So instead of just looking at localization, which we've done, we can look at release. So this is the work of a postdoc in my lab named Jocelyn. And what we found is that when we look at the localization of these vesicles in different types of neurons within the endogenous expression pattern, we do find that they're trafficked to different subsites in different types of neurons. So that's cool. And what we want to do is correlate that with release. And at the same time, we're using FACS sorting, actually, to sort out these subpopulations of vesicles and look for markers, protein markers by mass spec, that would allow us to have this genetic access. And all of these experiments are being done at very high resolution imaging in a classic worm chip where the worm is actually trapped. But this allows us to look at 100x and to see things like vesicle release. The other questions that we're really interested in are really biophysical in nature. And so we're setting up a system where we can trigger neuropeptide release from a single point source and then image the downstream receptor expressing neurons using genetically encoded second messenger sensors. So we're starting with GCAMP, but you can imagine using voltage sensors or other sensors. And the idea is that if we specify the site of receptor expression, we can ask very specific questions about distance and timing, at least over the scale of a millimeter, which is how long the worm is. But that's a pretty long distance in the brain. And so what we want to do is ask, how far can a peptide travel to signal? And in the endogenous expression pattern, if we have two neurons that express the receptor that are equally activated or differentially activated by the peptide, does that have to do with the concentration or the timing, or is it some combination of both of those things? Or is there something else involved here, like, for example, specific expression of a neuropeptidase in one neuron and not the other? And would that give us some sort of specificity in signaling that, that we can't actually see right now? And this is all going on in microfluidic devices where the worms can move around freely. So these devices allow us to correlate behavior, at least as far as movement is concerned, and possibly mating, with a chemical stimulus if we'd like, but also with neuronal activity. And this is all work that was, these devices were built by Dirk Albrecht when he was a postdoc in Corey Bargman's lab, and we collaborate with him now. I actually don't know how much time I have left. Okay, good. This is one of my favorite things to talk about. Coming back to this cartoon of the synapse, classical small molecule neurotransmitters are obviously very quickly recycled or degraded, in the case of acetylcholine, by enzymes that are located here. And this work has, there's hundreds of papers over decades really working this out in beautiful molecular detail. for neuropeptides, we really don't have the same sort of clarity. And in fact, so neuropeptidases are thought to be inactivated by these, neuropeptides are thought to be inactivated by neuropeptidases, which can either be secreted into the extracellular space, or more often they're transmembrane proteins that have their active site facing the outside of the cell. And basically related to what Evan was talking about, the way that people discovered these neuropeptidases is by grinding up tissue and looking for peptidase activity in vitro. And so this has given us a list of about a dozen enzymes that can degrade neuropeptides in vitro. They tend to have very large substrate specificities, but we don't know anything about, and by isolating them that way, essentially we've thrown out any information about localization, which, with a receptor, with a membrane-bound neuropeptidase, that could be very important for signaling. And we think that because there's hundreds of peptides, and because the signaling is so important for so many different processes, that there must be specific mechanisms to inactivate them. So what we're doing is we basically did a search, a bioinformatics search, for elegans predicted peptidase genes. And we narrowed down a very large list to a reasonable list of 139 candidate neuropeptidases. And we threw out things that were involved in development. We looked specifically for things that had human homologs. And the goal here is to generate a map, to take all of these genes and to profile them by RNAi in the worm, and to look at what their endogenous substrates are. So this builds on what I did in Corey's lab. So we can use something that's related to Silac, which is called Silan, to differentially label control versus RNAi populations for each of these genes, and profile by mass spec all of the peptides that are produced, and we can ask which ones are changing. And this will capture not just which ones are changing when we knock down a neuropeptidase, but it'll also capture the breakdown products, which sometimes can have biological activity. And so we started to do this for, I guess, four neuropeptidases. And what we found so far is a broad specificity of things that are changing. But the goal is to characterize all of them, then to look at their localization, to ask whether or not they're actually providing some specificity to these signaling systems, and then move into testing their functional roles by manipulating their expression levels and seeing if that changes the signaling in our setup. So I'm going to completely change gears now and talk about a project that's relatively new in the lab, but that I think is one of the most exciting things we're doing. So in thinking about how to translate our work into mammals, which is really the goal, I feel like all of the experiments that we've done, that we are doing in worms, are appropriate to do there, because at least in the context of a whole brain, you can do anything in slice, but in the context of a whole brain, those questions can really only be answered in a system like the worm right now. But we really wanted a way to manipulate neuropeptide signaling in the mammalian brain that would get around a lot of the problems that have clouded this field for a long time, namely that you get a lot of developmental compensation if you knock out a neuropeptide genetically from birth. There's a ton of functional redundancy built into these systems. So initially what we wanted was a way to knock out neuropeptide signaling acutely in a specific population of cells in the adult brain. And when we started thinking about it, all neurons express multiple transmitters, usually a fast and a slow or multiple slows. And we started thinking about oxytocin neurons. So these are neurons in the hypothalamus that they're called oxytocin neurons. But they also express 9 other neuropeptides, possibly more, and glutamate. And so when we started to kind of wonder what all of these behaviors that oxytocin has been ascribed to, either by pharmacological or genetic manipulations, we wondered how could we possibly figure out which of these neurotransmitters is actually important, or if any of them are, aside from oxytocin, for the behavioral outputs of these circuits that people are characterizing. So we came up with a method to manipulate individual modes of neurotransmission that satisfies 3 criteria. So that's cell type specific, that's acute, so we can do it in the adult, and that's directional, meaning we want to be able to silence neuropeptide output or input, or really any class of neuromodulation in either direction. And our approach is based on implementing CRISPR-I in the brain. So for those of you who are familiar with CRISPR, CRISPR has 2 components, obviously. A Cas9 enzyme that binds to the DNA and cuts it, and a single guide RNA that programs where that protein is going to bind to the DNA. So CRISPR-I is analogous, I guess, to RNAi in that they've taken the dCas9. This is work from Jonathan Weissman and Stanley Chi at UCSF. They took the dCas9 protein and removed the nucleosides. And in doing that, the Cas9 can still bind to the DNA, but it can't cut it. And they fused that to a transcriptional repressor, in this case CRAB. And if you use an appropriately designed guide RNA, what they found is that you can target this protein to any site around the transcriptional start site and block transcription. And what's beautiful about this system, what's really very powerful about it, is that unlike RNAi, which uses the endogenous protein machinery of the cell, this is completely orthogonal, and it also is much more specific. So the reason it's more specific, at least one reason it's more specific, is that you have to target the guide RNA to a window that's plus or minus 50 base pairs around the transcriptional start site. So what that means is that the universe of possible off-target effects is limited to the 20,000 or so transcriptional start sites in the genome. And this is data from Jonathan's lab showing that it's very specific in human cells. And so the goal here was to implement CRISPR-I so that we could acutely knock down transmitters in the adult brain. So there's 2 components to the system. There's the dCas9 CRAB. And so we took several approaches to do this. One was a knock-in mouse, which expresses the dCas9 CRAB protein everywhere. And the other is viral-mediated approaches. And so we've actually translated this to HSVs as well. So AAVs and HSVs will express on different timescales. So depending on what the experiment is you want to do, you might take advantage of either one of those. And of course, the second part is a single guide RNA. And we needed to come up with a way to make the cell type specific. So we made the guide RNAs CRE-dependent by borrowing a trick from the shRNA field. So they took a U6 promoter and modified the TATA box to include a lock site, so that now the guide RNAs are CRE-dependent. So what we can do with these is then inject them into any CRE line of interest and combine that with any number of neuroscience techniques, including optogenetics, if we want to look at optically evoked behaviors, and silence individual modes of neurotransmission. So for those oxytocin neurons that I was talking about earlier, this is a list of all of the guides, the viruses that we've made so far. And so for oxytocin neurons, we wanted to silence oxytocin itself, glutamate output, or all neuropeptides to ask what were the relative contributions to different behaviors. So in order to silence the neuropeptides, what we did is we found that if you target a combination of enzymes that are required for neuropeptide biogenesis, so these are the enzymes that are required to cut the peptide out of the precursor and then get it packaged into the vesicles and released. If we knock these out combinatorially, we can essentially knock down all neuropeptide signaling from just those neurons. And this is a short list of the things that we've made so far, but you can imagine extending this really to any transmitter, any circuit. So the key here is validation, obviously. So one of the lovely things about CRISPR-I, compared to CRISPR, at least in the brain, trying to validate whether or not CRISPR is working acutely in an adult is very challenging. You can stain for things, but there aren't antibodies against a lot of the proteins that we like to knock down in the brain. So with CRISPR-I, what we can do, since we're affecting the RNA levels, is we can use sequencing and qPCR. So the first thing we did was to just look in places where we have intermingled cell types with very similar, these genes are actually very similar, to look at the specificity of our single guide RNAs. By looking at intermingled cell populations. And I should say that we made an in vitro system so that we could first screen the guide RNAs before we put them into the brain. And then we can look at the efficiency of silencing by doing things like injecting the virus, unilaterally into a bilateral structure like the hypothalamus, and ask whether we get specific knockdown on the side that has the virus compared to the contralateral side. And so this is data from an oxytocin single guide RNA, one of the early ones that we did, where I think this was one of the first places where we saw a knockdown, so it was very exciting. And all of this work is being done by a postdoc in my lab, Kristen Obrakta, in collaboration with the Knight Lab at UCSF. So the other way that we're validating is using a ribosome tagging strategy. So the idea here is that we can either incorporate a GFP L10 tag, so we're tagging GFP on one of the ribosomal subunits. We can either incorporate this into the single guide RNA vector, or we can inject these separately. And so when we do this, we get all of the cells that express CRI are having their ribosomes labeled by GFP, so you can immunoprecipitate them in sequence. And at the same time, we inject a virus that has a different tag on L10 that is not CRI, it's not floxed, so that those neurons that get infected by this virus will have this orthogonal tag, so that we can then sequence both populations. So we can see around the site where we infected whether or not we got very specific CRI-induced recombination. So this is some preliminary data from an AGRP CRI line where we injected these two viruses. And you can see that in the GFPL10 IP that we very much enriched for MPY and AGRP, which is what we expected to see. So this is all nice. And this is all ongoing work. So thank you for listening. This is the view from my office window. If anyone ever wants to come and give a talk at sunrise, actually. And I have to thank a lot of people. Corey, I was very fortunate to have Corey as my postdoctoral advisor. I learned an incredible amount from her. And these are the people who were in her lab at the time who helped me with the Nematocin project. And in my lab, I talked about work by Kristen and Jocelyn. And Courtney is a technician who helps Kristen. And then we have a lot of collaborators. Many of our collaborators are for projects I didn't talk about today. But at UCSF, we collaborate with Zachary Knight, and Gwendolyn Daly in her lab, and that lab does a lot of injections for us. And Michael McManus made the Cas9 crab mouse that we're using. And Brad Gibson is a proteomics specialist at the Buck who's helping me with all the mass spec data we're generating from the Neuropeptidase project. So that's it.
We have time for a few questions.
Hi, I like your talk. So I have a small technical question. So regarding the fluorine that tag the Nematocin.
Nematocin.
So yeah. So is it possible to visualize the single quanta relays?
The single what?
Is it possible to visualize the single quanta relays, the presynaptic quanta?
So, no. Well, I guess if you were to, no, we haven't seen that. So we're looking at the signal to noise is pretty high. So we get like a 5 to 10% delta F over F. I can show you some of the data if you want.
Okay.
Yeah, but
great.
we don't see single quanta release,
Yeah.
at least not with the imaging setup that we have here.
Because the expression level is low in...
It's not so much that it's low, it's just that the vesicles are really small, and compared to mammalian vesicles, the neurons are small, the vesicles are small, everything's small in C. elegans.
Okay,
It's one of the drawbacks.
I see. Thank you.
Question down here.
So in terms of the biophysics of the release in either C. elegans or mouse, if a neuron has multiple neuropeptides, do they get released at the same site? And is it ever differential release, or is it always at all 7 or however many?
No, not at all. They're always, I think that in most cases they're differentially released. So there's data from in vitro cell culture systems in human cells showing that within the same neuron, if you tag different neuropeptides, you'll find them depending on the neuron, you'll find them in separate vesicle populations, and sometimes you can find them in the same vesicle populations. So there's a lot of in vitro data that suggests to us that there are these multiple populations that are of vesicles that are functionally distinct. And in worms, so we've only looked at nematocin. There are some other peptides we can tag. It's hard to tag peptides usually because they're processed on both termini, but there are a few that aren't processed on the C terminus that we can tag. But with nematocin, what we see is that we see the vesicles traffic to different sites in different types of neurons. And in one case, you can see it within the same neuron that we have vesicles in different places. So we're still tinkering with the florin. But once you get that working, the idea would be to try to see under what context do we see things released at different places.
And in mammals, is this release always at axons, or can it also occur from dendrites?
It can occur anywhere. So it can occur at synapses, and oftentimes when you see dense core vesicles released at synapses, they are co-packaged with the classical neurotransmitter. But you can see, I actually don't know if you can see release. I think you can infer release by doing microdialysis and things like that. But you can see the vesicles localized in dendrites and also in the soma. So, yeah.
And then one last question. Aren't you still, in terms of, because you asked the question, how far can they reach? I mean, in terms of timing, they're still limited. The transport, once they're released, is via diffusion, right? So they're still limited by square root.
I mean, if you do the math, that doesn't make any sense, right? It can't be just basic diffusion. There has to be something more complicated going on. And maybe it's just the tortuosity of the space, right? It's not just a glass of water that they're diffusing through. But yeah, we really don't know.
So it's not known whether it's only passive diffusion?
No, we don't know. And I don't think that it can be just passive diffusion. Maybe it has something to do with the 2D transport, along a surface or something.
I mean, given the time scale of this behavior, which is over minutes, and given the tortuosity, it could still be effectively one-dimensional diffusion.
It could be, yeah, it could be. And maybe I don't know. The answer is, this is what we want to know, yeah.
A question in the back.
With regard to the modulation of learned behaviors, perhaps more complex behaviors, especially in mammals, what's known about the plasticity of the receptors for these different peptides?
So there's a lot known actually, and it's very specific to each individual system. One thing that I didn't mention about peptide signaling is that oftentimes you'll see autoexpression of the receptor. So oxytocin neurons, for example, express oxytocin receptor. And what happens is a feedback loop where essentially oxytocin will feed back on the neuron and induce this process called priming, where the vesicles move from a non-releasable state to a very readily releasable state. And suddenly this neuron that only responded to a low sustained level of calcium before, now that it's been primed by that first initial release, will respond to different types of stimulus. But in terms of receptor desensitization and things like that, it definitely happens. It's just very specific to the different individual neuropeptide signaling systems. Does that answer your question? Okay.
I was wondering if you could speculate on the mechanism whereby you lose this peptide and end up with this behavior. So you describe motor fragmentation, then you describe sort of not able to find the target. But it's not entirely clear to me how the loss of the peptide leads to those phenotypes.
So we didn't do, there are experiments you could do to actually figure that out. But what we think is going on is that the peptide is feeding back on those neurons that feed into DVA, the ones that control, the sensory neurons that control, how light touch is interpreted, for example. And they're also obviously signaling to the neurons that are in the tail that are doing all these specific motor programs. So what we think is that the peptide, you can list the possibilities, right? It's either changing the gain or it's changing the sensitivity of the neuron to different stimuli, or it's turning it on or it's turning it off. And we haven't checked. But what we think is going on is that it's somehow changing the properties of those neurons to basically make it so that rather than funneling everything towards this motor program that now suddenly the context is lost. Does that make sense? I'm sorry, that's not very satisfying.
All right. Oh, wait, we have another question.
Probably the last question. I wonder, actually, is there any way to systematically quantify the distribution, the concentration of the neuropeptides across the entire worm?
Well, you can look by mass spec, right? So you can do quantitative mass spec. The problem with that is obviously you need a very large amount of material depending on the expression level of the peptide. You can indirectly infer it by using, a promoter, a transcriptional fusion, essentially. You could try with peptides that can be tagged directly. You could try to quantify them by tagging them directly, but those are the only ways that I can think of to measure. Worms are too small to do things like microdialysis.
All right, thank you very much. And thanks to all of our morning speakers. We're now going to break for lunch. Lunch will be served on the cafe in the 6th floor. If you go up the elevators and turn right.